Section 4, we'll continue looking at the digestive system by looking at the small intestines. First of all, the functions for the small intestines. So most everybody realizes that's where your food is absorbed, your nutrients and water is absorbed from the food that you ate and digested into the bloodstream. But there's actually a lot of digestion that takes place in the intestines, and that's because of some enzymes that enter into the intestines from the pancreas, and we'll talk about those enzymes in just a little bit. Now, there's three main divisions of the small intestines, beginning with the duodenum. You kind of see that on this image here. You have the end of the stomach here. This is the pyloric region of the stomach. There's a sphincter here, or a muscle here that kind of helps control, and a valve here that helps control the release of the digested food, or the acidic chyme that we mentioned, helps control that going into the first section here called the duodenum. It's about 25 centimeters, so it's relatively short, about 12 inches long, relatively short. And then you have the second section, which is much longer, the jejunum, which is roughly 2.5 meters, and then the ileum is the final section, the last section, and that's what connects the intestine, the small intestines, with the large intestines. Now, to help in the process of absorbing all of those nutrients that you digest back into the bloodstream, there's several modifications we'll talk about that help to increase the surface area on the inside of the small intestines. So, for example, if you were able to look at the small intestines here, and you're able to look on the inside, instead of being just smooth on the inside, you've got several layers or several types of modifications that help to have expose more surface area. So in this case, more surface area means more space for absorption of all these nutrients back into the bloodstream. And so instead of being just smooth, first of all, you see just a folded region. These are called circular folds, and that adds some more surface area. Each of the folds have kind of a finger-like projection that kind of stand out like this. These are called villi, and this is where your capillaries are at. Those capillaries are there for exchange, and in this case, they're there to absorb all those nutrients into the bloodstream. So this is your connection to the circulatory system here. And then on the edges of all of the villi, you have little small projections, little very fine thread-like projections called a microvilli. And so all three of these together help to increase the surface area. Your book estimates about 600 times. So you have 600 times more surface area and more ability to reabsorb all these nutrients than you would if it was just smooth throughout. Now, some of the secretions that you'll find in the small intestines, of course, there's mucus, but there's mucus, throughout the GI tract. Mucus is there to help, again, protect, in this case, the small intestines from any enzymes and from the stomach acid that's leaving the stomach and going into the intestines. So you got to protect the small intestines as well. So you will find mucus there. There are enzymes there that are coming from the pancreas, and some of the examples of enzymes here are disaccharidases, peptidases, and nucleases. First of all, if you don't already recognize this, if the word ends in the ending ASE, ACE, that usually means you're dealing with an enzyme. And so a disaccharidase is an enzyme that helps to break down disaccharides or break down sugars, such as sucrose. Sucrose is a sugar that we eat that's in our foods. If you have to have an enzyme to digest that sugar, and so you would need a disaccharidase, this enzyme, to break down sucrose. Peptidase, this is what's needed to break down peptide bonds. Of course, you find peptide bonds in proteins, and so you need a peptidase and a special enzyme here to break down proteins. And then DNA, RNA, those are nucleic acids, and so you have specialized enzymes called nucleases to help you break down the DNA and RNA. Now, connecting with the small intestines would be the large intestine. And the main function here for the large intestine is also absorption, but it's mainly absorbing any remaining water, maybe not all the water, but most of the remaining water and any salts or electrolytes or ions from the food that's passing through. This is now, you're no longer really absorbing any nutrients. So the small intestines have absorbed as much nutrients as they could. You've completed digestion now, and so anything that you're not able to digest is now passing into the large intestines. So this is reserved for things that you do not have the ability to digest, and so and you're reabsorbing any water. You don't want to lose any water if you don't have to. You want to conserve the water, conserve your electrolytes, your ions, and your salts. You don't have to know necessarily these numbers, but it can kind of give you an idea. Roughly about 1,500 milliliters of the acidic chyme that we talked about, from the stomach going into the small intestines. About 1,500 milliliters of that on a daily basis goes into the first section, goes through the cecum, which is your this first section here, enters into the first section there of the large intestines. And about 90% of that actually gets reabsorbed because most of that is fluid, most of that is water. And so what doesn't get reabsorbed is excreted as solid waste. That makes up about 80 to 150 milliliters on a daily basis. So just a few sections here to kind of mention some of the anatomical terms, the anatomy of the large intestines, the cecum, that is the first section there. Just off from the cecum, if you look at the diagram, figure 1621 here, this is from a different textbook, so this is not the same as in your textbook, but the first section there, that's also where you're going to find the appendix coming out. Here's the cecum. Here is where the small intestine, the ileum, is coming in. And so there's a valve here called the ileocecal valve. That valve helps to prevent any backward movement of material from the large intestine back into the small intestine. So you do have a valve there. So the first section here is called the cecum. The main portion of the large intestines is referred to as the colon, and it really contains four main areas. The ascending portion of the colon is here. The transverse means it goes to the side or kind of horizontal, and so that goes across. The descending portion descends down, and then you have a sigmoid region, which kind of means S-shape. So you have a sigmoid region that will eventually open up into the rectum, which is just a straight tube that connects the colon to the anal canal. Now, there are many good types of bacteria that we have in the body. Of course, there's many pathogenic or disease-causing bacteria that could cause problems, but there are many good types of bacteria that reside in the large intestines. And as long as their population is kept under control, and as long as they pretty much remain in the large intestines, then everything, your balance of bacteria usually does fine. Some of the benefits of these bacteria, number one, is sometimes these bacteria can produce some vitamins that they don't need, but we can actually use them. And so, for example, vitamin K, this is a vitamin that we need. It has several reasons, several uses. One is for blood clotting. You need vitamin K, and it turns out that some bacteria, some E. coli type bacteria that normally reside in the large intestines, actually produces vitamin K as a byproduct, metabolic byproduct, and we can actually harvest that and use it for our own benefit. Sometimes you do have some enteric type bacteria that lives in your gut that help to digest some of those carbohydrates that we can't break down. And so, we call that fiber. There are some carbohydrates that plants produce, like in the cell walls of plants, that we don't have the ability to digest, but these bacteria do. And so you can actually have some bacteria to kind of help in the digestive process. The other thing is, if you have these good bacteria in place, they help to slow down and deter the growth of the harmful bacteria. And that's because they are actively competing for the nutrients, and they're competing for space. So they can actually be very beneficial at preventing the overgrowth of the harmful type of bacteria. Now, those are the main structures associated with the GI tract, but there's some accessory structures, organs that we need to talk about that's part of this system. We've already mentioned the pancreas in an early chapter back in 16 because it is an endocrine organ because it does produce hormones. Well, it also functions as an exocrine organ because it does have, it is a gland that produces enzymes. There's specialized cells called acinar cells that produce digestive enzymes They exit the pancreas through a duct, a pancreatic duct, and in the diagram you kind of see that duct. So the gland cells produce the enzymes, it funnels into the ducts, then those ducts lead over to that first section of the small intestine. So you have an opening here in the intestines where you have the pancreatic enzymes or pancreatic juices going from the pancreas into the small intestines. There's also a bile duct. We'll talk about bile in a minute when we talk about the liver, but there's also a bile duct that comes down and joins that location as well. So that bile from the liver, from the gallbladder area, goes down into the small intestines as well. So let's look at those pancreatic secretions or juice or enzymes that are part of that are excreted from the pancreas going into the small intestines. This is an aqueous-based solution, so it is water-based. You also find ions there. Sodium ions are there, potassium ions, and bicarbonate ions. And so bicarbonate is a base, just like baking soda is sodium bicarbonate, and so you actually are producing a bicarbonate type ion in the pancreas that helps to balance out the pH, helps to increase the pH. So, in the stomach, you're producing hydrochloric acid that has a very low pH, somewhere around pH of 2, and that's needed to digest the proteins and activate the pepsin inside the stomach. But when you move the acidic chyme from the stomach into the intestines, you need to change the pH, because most of the enzymes in the intestines need a more neutral type pH. They need a higher pH. And so to help change the pH, you have bicarbonate, part of the pancreatic juices or pancreatic secretions, coming into the intestines. It helps to balance out the pH. So these are some of the enzymes you would find there. Trypsinogen, that's there to help digest the proteins. That's different than pepsin, because pepsin works at a low pH. This is working at a more neutral or a little bit higher type pH, somewhere around 7 or 8. Pancreatic amylase, this is helping to digest starch. You actually have a version of this produced in the mouth, and that's called salivary amylase, because it's produced by the salivary glands. You can actually begin digestion of starch in the mouth. Pancreatic lipase, that helps to digest lipids or fats. And then again, you have the deoxyribonuclease and the ribonuclease that helps to digest the nucleic acids, DNA, and RNA, because the food that you eat, doesn't matter if it's plants or animal-based or a combination, is going to have DNA and RNA, and you need to be able to digest those and break those down. Another very important accessory structure is the liver. So we need to kind of look at this. There's 5 main functions for the liver that we'll talk about. Number 1, we mentioned before, is bile production. Roughly about 600 to 1,000 milliliters of bile being produced on a daily basis, roughly. And in addition to bile, there's also some cholesterol and some fats or lipids and even some hormones that kind of goes into the mixture of the chemical we call bile. And you see some of the functions up here. We've already mentioned the second function of emulsification of fats or lipids. That just means it kind of breaks them down into smaller little particles or smaller droplets. But it also helps to balance, again, the stomach acid. So this is going into the small intestines. The bile is going to go to the small intestines, where it's going to help to neutralize and help dilute down some of that stomach acid. Number 2 function of the liver is storage. And so you can store many nutrients. You can store glucose in the form of glycogen. You can store lipids there. You can store vitamins. And even some elements such as copper and iron can be stored in the liver. Number 3 is very important. This is nutrient interconversion. You can convert nutrients from one form to another. So for example, you can turn something like amino acids into a compound that's needed for energy, because normally amino acids aren't needed for energy. Usually amino acids are there to build proteins, and proteins are for structures or enzymes or other functions. But here in the liver, you can actually transform chemically, do a chemical reaction with amino acids, and actually transform those into something like ATP or glucose. Part of the vitamin D production process takes place in the liver. Now, of course, a lot of that begins in the skin, and you get some UV light from the sun. That helps to convert some molecules over in the skin that are needed to make the vitamin D. but some of that process also takes place in the liver. Function 4 is very important. This is probably why the liver is most well known, I guess, is because of its detoxification properties. It helps to make harmful chemicals safer. It helps to detoxify harmful chemicals, whether it's natural chemicals in the body or if it's chemicals you're putting in your body. So, for example, hepatocytes, these are liver cells. They help to break down metabolic waste products. So when you digest proteins, you're producing things like ammonia, and that's a harmful product. It's toxic to the cells, and so you got to get rid of it. And so one of the things that the liver can do is to detoxify the ammonia, convert it into something less toxic, such as urea, that gets filtered out of the blood by the kidneys and will be excreted, out as waste. But also things that you put into your body. So, alcohol going into the body, that's a toxic chemical. It has to be dealt with in the body. And so the liver is going to try to break that down chemically, or medicines, or, acetaminophen or Tylenol or ibuprofen or antibiotics. All of these chemicals have to be processed somehow. And it's the job of the liver to eventually detoxify any of these medicines or chemicals or drugs. The number 5 is excretion. There's some waste products that actually get excreted, and that goes through the bile. And one example is bilirubin. This is a pigment that is broken down, is left over from the breakdown of red blood cells. So after red blood cells are used up, they last in the system, 3 or 4 months. After red blood cells are broken down, some of the pigment that's inside of them, called bilirubin, that gets processed by the liver and gets excreted through the bile system. And then make a note of the gallbladder. This is a storage sac for bile. So don't get that confused, that bile is actually produced by the liver, but it's stored and concentrated and then released from the gallbladder. The gallbladder is located just underneath the large lobe here of the liver. And there is a duct here that runs from the gallbladder down to that first section of the small intestines we mentioned a while ago, called the duodenum. The last section we'll look at, digestion and absorption. It kind of goes through the processes again of digestion and absorption and transport. Of course, digestion, I think you realize, is the breakdown of food molecules. That is both mechanical and chemical. Mechanical just means you're breaking it down, you're chewing it up into smaller pieces. That could also include the emulsification process, because that's really mechanical. You're not chemically changing fats during emulsification. You're just making them smaller. Chemical is anytime you chemically change something. You're chemically breaking a bond in order to do that, and you have to have some kind of enzyme or acid or something to help chemically change something. Absorption and transport, by definition, is just moving those molecules out of the digestive tract, getting it into the bloodstream. Of course, that's really the job of the intestines, small intestines, and large intestines, if you include water there as well. And then just a quick review of some of the major biomolecules that are used as nutrients in the body. This is, you should have seen this back in 168, probably around chapter 2, when you talked about some of the organic chemistry there, the biomolecules there. So if you need some refresher on some of these molecules, go back and look at chapter 2. Towards the end there, it talks about the carbohydrates and lipids and proteins. Carbohydrates, we think most of the time we think of as polysaccharides, although there are monosaccharides and disaccharides. Those are your simple type sugars, but you also have polysaccharides. These are your complex carbs, and that includes things like starches and sugars. We digest those beginning with the mouth, because we have that enzyme we mentioned just a minute or so ago about called salivary amylase, and that's produced by the mouth and actually helps to start the process of digestion. Then the digestion of carbohydrates continues in the stomach, and then it is finished in the small intestines because of the pancreatic enzyme, pancreatic amylase, and then some of those disaccharidases that we mentioned earlier. Lipids, there's many different forms of lipids, and you can look back at chapter chapter 2, and we're going to talk more about these nutrients in the next chapter as well. But these are examples of lipids you find in the body. Triglycerides, those are the lipids that many times you test for or are tested for. If you go get some blood work done, a blood panel done, you're probably going to get a number for your triglyceride level, your fat level in the blood. Phospholipids, these are very important. You find these in cell membranes. Steroids, these are common in the body. That includes cholesterol, but it also includes certain hormones like your reproductive type hormones, testosterone, estrogen, progesterone. These are all steroid-based hormones. And then some fat-soluble vitamins would fall into this category as well. Now, you may have seen the terms HDL and LDL. These are actually lipoproteins. Most everybody relates these to cholesterol. These technically are not cholesterol, but they are usually connected to cholesterol. And so LDL is low-density lipoprotein. It's low density because it has more lipid attached to it and less protein attached to it. And so that its function is to transport cholesterol out to the cells. Now, if you go to get a checkup done, your LDLs, this is what a lot of doctors are going to call the bad type of cholesterol, because it's needed. But if you have too much cholesterol in your diet, in your system, then sometimes these LDL proteins will actually deposit some of that cholesterol out into the bloodstream. And so you don't want that to happen. You don't want the cholesterol to build up in the arterial walls. And so you need the cholesterol. You need a certain amount of it. Cholesterol is needed for cell membranes. It's needed to build, make some of the hormones we mentioned. It's part of the bile. I mentioned that already. But, too much, you can have some problems there. So you have to monitor your LDL number. Your HDL number, this is high-density lipoprotein. And that's because there's less lipid there. So this is more dense because of the more protein. Proteins are more dense. And so this is why this one is high-density lipoprotein, because you have a higher amount of protein there than the other one. This is a lot of times called the good cholesterol. And that's because this is trying to kind of get rid of some of the cholesterol. So it's actually taking extra cholesterol out from the cells that the cells don't need and taking that back to the liver for storage and for getting rid of some of that extra cholesterol. So you need that HDL cholesterol to help kind of clean out some of that extra cholesterol you don't need. Proteins and protein digestion. You have to have an enzyme to break down proteins, and pepsin is that main enzyme. Of course, pepsin also requires acid to do this. That's also why you have hydrochloric acid in the stomach, is to help activate the pepsinogen turns into pepsin in the presence of HCl or hydrochloric acid. And you have enzymes, proteolytic enzymes. Those are just protein digesting enzymes. They help to break down proteins into the amino acids.